And I brought a laser pointer, so it should make it a little bit easier for you guys. Don't let me forget this either. Okay. I'll make sure this works.
Great read. Yeah, that's kind of nice.
It's like fancy, you know? Okay. Nope, you're fine, Jeffrey. Just wait. I know you're anxious. So, okay. The question has already sort of been raised about, well, how stupid Canvas is, and I totally agree that, because I spent some time trying to figure out how to work this so that when you saw your grade, it was actually reflective of your grade, and it's not, because Canvas is stupid. So I did go through, those of you who did the extra credits, okay, you were provided with those points. But the way Canvas works is say, take a look at that as an individual assignment and provides and calculates the grade based on that, which again is stupid. So to quickly determine what your grade is simply add up all of your points and then divide it by 200, and boom, you have your percentage. You can multiply that by 100 if you need to figure out the total numbers. But you do that and you can determine what your grade is at this point. So it's all the points that you've earned, including the extra credit ones, and then just divided by the 200, which is the total number of points that are available at this point in time. So, and again, Canvas. So, but, okay. Any other questions or concerns? Okay, awesome. So, Jeffrey, want to come on up? And then after Jeffrey's presentation, Gabe will go over his article. So, and then to use this, the pointer is up top here. And then to go forward, simply up, backwards, and don't hit this button because it'll give you a blank screen. Okay, okay, there you go, Jeffrey. And speak loudly so everybody can hear you. And I'm going to say that to everybody. Looking at you, sir. Okay.
All right, good morning. My name is Jeffrey, and my NSF proposal is going to be about microplastic studies in the microbiology ecosystem. So, plastic is a ubiquitous environmental pollutant. This plastic is found in several ecosystems. Like you can see, these are like this is the ocean, and then this is like a landfill, right? So we have plastics in the rivers, in the water bodies, and then in the land. Now, between 1950 and 2017, we produced about 9.2 billion tons of plastic, and we've only incinerated about 1 billion tons of this plastic that we produce, and about 5.3 billion has been discarded. So where do we discard all this? We discard this in the river bodies and in the water bodies and in the oceans and then in the landfills. But then experts say by 2050 there will be more plastic in the oceans than fish, right? Because if you continue depositing these plastics in these oceans, then it means that these fish and these marine organisms will be feeding on these plastics. Now, we can categorize plastics into different types based on the size. So, if you have a plastic less than 5 millimeters, then you can say this plastic is a microplastic. So microplastic generally bioaccumulates in the environment. So you can see humans are the biggest culprits here. So we litter plastic all over in the environment. In the industries, they keep producing more plastics. Now these plastics can biodegrade in the environment by UV and mechanical stress. We produce more microplastics, and these microplastics are again ingested by these marine organisms like fish, etc. And then, of course, these bears also feed on these fishes in these water bodies. So we keep transferring the microplastics, we put them in the ocean, the fish is degraded, and then these higher trophic organisms also keep feeding on it. Now, it has been estimated that humans eat over 40 pounds of microplastics in our entire lifetime. You might not see this, but several research has shown it. Now, those of you who like seaweeds, now it has been found that most seaweeds are usually sequestered with microplastics. So the more of these food that you're eating, you probably are also consuming more plastics. Now, Blanc Mittal conducted a study, and then they found out that inhaled micro and nanoplastic particles may pose higher risk of cancer in the future. And microplastics can also serve as vectors for pathogen transmission. Now, when we say vectors for pathogen transmission, we mean microorganisms and other dangerous chemicals can be sequestered on these microplastics, and then as these microplastics are being transferred to the trophic levels, we kind of we have these microplastics are made available to us, and then if there are any like pathogens, we can also get them. So this term plastisphere is like a general term used to describe like a diverse group of microorganisms that are living on a plastic surface. So when we have biofilm formation, or when we have like other groups of microbes living on the surface of plastics, we call this a plastisphere. So this is like a general overview of what a plastisphere is, like a microplastic interaction. So here we can see the microplastic fragment surface, right? And then we have biofilm formation. This is a vibrio species biofilm biofilm, and then these are like a multi-species biofilm. And then we can see different types of microplastics here. You have microplastic fiber, we have microbeads. Now, you can see microplastics, you can see biofilm formation on the surfaces of these marine organisms as well, right? And then we can see like heavy metal accumulation on the surface of the fragments of the microplastic, like aluminum, we have copper, and then we have zinc. And then, of course, we can also see this chemical here called DDT. This is a very toxic chemical which was probably used back in the day for fishing, but it has been banned by the EPA. But then these toxic chemicals can also be sequestered on the surfaces of these plastics. So you can see if this microplastic gets into your food or in your water, you are probably being exposed to all these interactions. So I chose this site for the NSF proposal because it's a very contaminated area. So back in the 1700s, there are a lot of like mines here, and these mines are like lead mines, right? So they used to mine lead in this area. And then these lead mine drainage drain from here into this big river. So this big river contains about 8 million tons of lead, right? And it's you can see here, this is Southwest Missouri Superfund National Priority. So this is a very huge problem being addressed by the Environmental Protection Agency. Now, the economic cost of antibiotic resistance to the US economy alone is an estimated 20 billion in direct health. And then many heavy metals augment proliferation of antibiotic resistance due to their antimicrobial properties, because metals in itself can serve as antimicrobial particles. So if you have an ecosystem where you have a lot of metals, there's a high chance that these microbes might also develop other resistances. Antimicrobial and heavy metal tolerance in microbial communities is facilitated by the plastisphere formation. So we saw all the interactions on the plastisphere, and we saw how these interactions can affect us. So my research objectives. Now, the first one, I want to determine the dynamic processes of biofilm formation on different microplastics submerged at varying depths and times in the big river. And I also want to determine the influences of biofilm formation on the surface morphology, physicochemical properties, and microbial community profiles on microplastics in the big river. my research hypothesis. Now, I hypothesize that biofilm formation of microplastics may increase with increasing depth and exposure time, and the biofilm reduces surface hydrophobicity of microplastics at varying degrees. And dominant microbial species might change with increasing exposure time and depth, as well as the type of microplastic. And then more than 70% of microbial isolates from Big River will carry both antibiotic and heavy metal resistant genes. So this is like an overview of my experimental design. We're going to use these 5 classes of microplastics. So we will acquire microplastics, polyamide microplastic, polyethylene terephthalate, and polypropylene, and polyvinyl chloride. So we expose these microplastics at this depth, so 2, 6, and 12 liters. Now, 2 et al. Published a paper similar, they did something similar, but they use PE microplastic, right? And then this is kind of a stainless steel cage, which is filled with the microplastics, and then they kind of use some varying weights. They put some weights at the bottom of the cage so that the whole thing will just like suspend, right, at different depth. And then the exposure time will be for 30 days, 60, and then 90 days. So we put this in the Big River, and then we leave it for 30, 60, and 90 days. So we will take our samples on these days. Now the size of the microplastic will be 4 millimeters, and the density should be approximately 1 gram per cent. Yeah. So once we collect our microplastics in the Big River, we bring it to the lab, and then we perform these microbial analysis. Now, for this part, we need to work on both the microplastic and the biofilm. So what you see here, these things are the biofilm on the surface of the microplastic, right? So we analyze the microplastic alone, and then we analyze the biofilm alone. Now, the microplastics here, we need to undergo what we call a FTIR to see if there are any functional chemical changes in the microplastic surface. So we take the virgin microplastic and then we compare it to the one which has been exposed for 30, 60, or 90 days. And then we check the surface hydrophobicity. So usually more plastics are hydrophobic, right? But then when you expose these plastics to a new environment where these microbes are kind of interacting with these microplastics, then we assume that the hydrophobicity of the microplastic might change. So we need to determine the change in surface hydrophobicity compared to the virgin microplastic. So basically we are done with this part right now. So we come to analyzing the biofilm. So we can extract community DNA from this microplastic. Sorry, from these biofilms, and then we target the 16S RNA gene that for hypervariable gene using these forward and reverse experiments. So once we're able to sequence the community DNA, we can find once we know the sequences, we can either use further analysis like maybe bioinformatics to see if we can find some sequence homologies in terms of what genes might be present in this community DNA samples. Now, we can also do an extra test where we kind of form something like a selective media where we use some like common antibiotics like ampicillin, vancomycin, tetracycline at 100 micrograms per ml. And then we also might add certain metals. We know that the Big River is heavily polluted with lead, so we know lead will be present. But we can also add other metals from the Big River, because I mean we assume lead is there, right? But then we also assume other metals can also be present. So we want to go this EDX analysis. This is energy dispersive X-ray analysis. Yeah. So this quantifies the elements on the surface of the biofilm, right? So if there are other metals like other heavy metals, this will give us these metals, and then we can add these metals to our culture, so that every microbe or every microbial species that can be resistant to both these antibiotics and these heavy metals, we can further select them as a resistant species, and then we do further analysis on them. Okay. Now we can also determine the total biomass, the total biofilm biomass by using this crystal violet stain, and then we measure the absorbance at 5 nanometers. This will quantify the total biofilm on each microplastic, because we believe that some microplastics might be more ideal for microbial colonization than the others. And then we can also quantify live cells, dead cells, and the extracellular polysaccharide secretions, the EPS sheets that might be formed on the surface of the biofilm. So we use these three different dyes. This SYT09 will stain live cells in green, and the propylium iodide will stain dead cells in red, and concanavaline A will stain the extracellular polysaccharide sheets. So expected results. Biofilm biomass on microplastics are increasing depth and exposure time will be significantly different due to changes in temperature, light intensity, organic matter, nutrients, as well as other environmental factors like dissolved oxygen, pH, etc. Surface hydrophobicity of MPs is expected to vary significantly due to the unique chemical composition. By the extent of chemical change, we don't know, will depend on the microbial community present. The dominant microbial species will vary with microplastic type. So we believe that maybe microbes that will dominant microbial species on PE versus PET might be different. And also the exposure time and the depth of the exposure also influence the type of microbial species present. And then due to the high lead concentration in the big river, we also expect that more than 7% of these microbial isolates might carry both antibiotic and elemental resistant genes. So in conclusion, microplastics provide ideal service area for biofilm formation, resulting in proliferation of diverse microbial communities. And we said these microbial communities share kind of information in terms of genes, right? Doing horizontal gene transfer. Understanding the interactions between global microbes and microplastics in contaminated ecosystems like Big River will provide more insight into developing alternative solutions to tackle emerging resistance chains. Thank you.
Questions for Jeffrey?
Can you go to slide 10 real quick?
Slide 10.
Yeah, that's the next one. Where does the greater than 70% of microbial isolates carrying antibiotic and heavy metal resistant genes, where does that 70% come from?
So we said that the Big River ecosystem is mainly polluted with lead, right? Right. So now, because it's mainly polluted with lead, the assumption is that the assumption is that microbes may carry other genes, right? So these genes may be antibiotic resistant genes, right? So it's like the metals itself may serve as like antimicrobials, right? So the organism might find a way to like pump these metals out of the cell, right? So if you have once they are pumping these metals out of the cell, they also might pump out like maybe antibiotics, right? So the most organisms that are resistant to heavy metals are also resistant to antibiotics. So if we have more organisms resistant to lead, then there might be other organisms, there might be more organisms which might be also resistant to antibiotics. So this is just something that can be expected, but it's also an experiment. So it might see that after the experiment, I might not see this.
Yeah, I didn't know if like other experiments had been done similar to this that like had a quantified value.
Well, I wouldn't say there's a quantified value,
Uh-huh.
but this is just something that I think might help.
Okay, thank you.
From a river mechanics standpoint, where are you putting your sampling devices? Because if you put it in something like an eddy where you have a circulation of water where there's like, it kind of sits there, or if you have it in a high flow environment in the thaw wag, or if you have it in a low flow environment, that could affect the different exposure limits of lead or other, like, or microplastics even, because you might have some areas that are low flow where the river kind of circulates the same water a little bit more, so then you'll have like a greater chance and concentration of what could be flowing down the river in terms of like microbes or lead. But then if you have it in a really high flow environment, it might be going by too quickly for it to actually like affect your experiments. Like, where in the river are you placing these? And I kind of understand the depth part.
Yeah, I didn't really think about this like a question in terms of where, like the exact part of the river. But I think we don't need like a high tide area. We don't need where like the river is like moving like so fast. Like we need, maybe I'll choose a site where the river is like probably like stable. Like I'm talking about like the current of the river is not that strong, probably. But I'll do more reading on this. We'll see this.
For your experimental design, why would you do it like immediately in the river first? Like, wouldn't it be better to do it like in a lab setting to like control for like temperature and light exposure, keep those constant to really see what effects?
Yeah, so like most researchers have done, most research on microplastics are usually done in the lab, right? Lab setting. But this is one of the challenges in this area of research because it's like we want to simulate what is happening in the laboratories in the lab, but sometimes the simulation is not that accurate. So it's like you don't get environmentally realistic concentrations, right? So whatever you're trying to simulate might not be exactly in the environment. That's why we want to incubate this in situ in the environment to see the actual effects.
So are you exposing, so you're gonna have the exposure depths at 2, 6, and 12. Are you going to have each of those be exposed, like have like 3 sets of those at 30, 60, and then 90 days as well?
Yeah.
So there you'll get all of the depths and all of like the exposure time, like combinations?
Yeah.
Awesome.
And we're going to replicate this about 3 to 4 times. So for each microplastic, we'll have like several of them. So this cage that you see here contains more than 200 pieces of this for each of the microplastics because I mean you're just cutting the microplastics into this size and just putting them in, right? So for each sample, so if you take this cage will contain 200 microplastics. So you can get a lot of samples to do one analysis.
Just have like a quick thought. That's obviously experimental, theoretical, but like maybe what would be the end like way of keeping extra microplastics from being introduced into the ecosystem? And do you have a way to just try and limit the amount that just gets swept away by the river?
No, honestly, I don't. That, this is just like trying to maybe find connection resistance strengths in this polluted environment. But I don't think anybody has like a lasting solution of how to
Curious, like, just to make sure you don't get any microplastics spread anymore.
So like for the 30, 60, 90 days, is there like a season that you're hoping to do this in, or are you hoping to do it in like multiple different seasons? Because in the wintertime, you're gonna have lower nutrient levels anyway and lower temperatures, and it's a drastic difference, especially in Missouri.
And I also didn't think about that. So I will incorporate that in my write-up.
I also have a couple of questions too. And actually, thank you, that was one of my questions. So, but I guess, first of all is, besides the heavy metals and all, what sort of impact is there on plastics in this particular environment? Is it known to have a lot of other anthropogenic input besides the redlining, the big river? So is it just because of the influence of the heavy metals, or because, another way of looking at this too is, what has been the impact of plastics on the microorganisms in this environment?
Yeah, so actually, I don't think any other work has been done on in terms of like what microplastics might be present there. I chose this site because I felt like microplastics are everywhere, right? Microplastics are polluted in all our water bodies, right? So if you have a heavily polluted ecosystem in combination with these microplastics, it might be very like detrimental, right? So that's why I kind of chose this to see if the microorganisms in this environment, how they will interact with these microplastics.
I want to include something about what are the overall anthropogenic impacts on this environment, and just to have sort of a background besides the metal influence. And then, what sort of environmental parameters do you have for this particular river? Because one of the things that I was thinking of, along with what Josh was mentioning earlier, is where exactly you'll place these devices to be exposed. And let me figure, I don't know anything about this river, so what is the depth of the river and how much, and then following up about the seasons and things like that, you really do have to provide a good understanding of the different environmental parameters. I mean, because when I'm thinking about 12 meters, that's going to be like 36 feet. Is the river even that deep?
The river is about it's more than 100 meters deep. Yeah, the river is more than 100 meters.
Okay, so you might want to actually, in your report, provide sort of a map of the river itself and some of these parameters. So, yeah, Josh.
Something also to consider is the proximity to historic lead mines that you want to put this in, because lead is so dense that even with like currents and things like that, like we find a lot of lead in the environment, especially in aquatic environments, being mixed in with the sediments. Since lead is relatively aqueous, it won't really just float through the water and hang out a lot of times. So what'll end up happening is, like, if you're really close to a historic lead mine, there might be some more mixed in at different stratigraphic levels within the river. But then as you go further down, you'll see more, like it'll start decreasing in value more and more. And there's going to be kind of these sweet spots where there's going to be heavy concentrations of lead, because that's the time it takes to settle down into the sediments, or like get washed up kind of in the banks and everything, actually, because it is so dense compared to the water and everything around it.
And then my other question is, how are you going to separate the biofilms, the microorganisms, from the microplastics that you can do the culturing and the DNA extraction that you're proposing to do?
Yeah, so usually with analysis of the microplastic, I think that is quite straightforward, right? You just take off the virus.
How do you do that? Are you going to scrape it? Or are you going to use a chemical means, or?
No, we're not going to use we're going to scrape off the biofilm, but we're going to.
Okay, so then that raises the question, how big is your microplastics?
The microplastics, you mean like the size? It will be maybe 5 by 5 squares. Yeah.
Okay, so we'll need to think about this. Okay. but yeah, and I don't know if physically screening it will work. So what you need to do is actually think about the individual steps. And so as we talked about, towards the beginning of the semester, just preparing samples and thinking about the assumptions you're making and, just to ensure that you have the best possible sample that you can have, which would be essentially, getting a good representation of all of the microorganisms that are present there. Jeffrey, this was really good. I mean, don't take all the questions as a bad indicator or whatever. You just want to know more, and it's a very good topic. So, okay, thank you, Jeffrey. Okay, and then there's something a little different. Kate is going to talk about the prokaryotic communities in latitudes. You can use that if you want to.
Great, hi. So, okay, we're going to be talking about these latitudes. Specifically, this is going to do with Microbiology Cavern in the La Palma Island in Spain. It has plenty of pseudoderms and colored microbial mats. So the overall study was that they took 4 samples to use with DNA next generation sequencing and field emission morphological characterizations. And they use those in order to study bacterial identification, functional profiling, and the morphological characterization. The data almost exclusively showed the dominance of bacteria over archaea. The distribution in phyla revealed that the majority abundance was protobacteria. In those samples, it was 37 to 89% in the 4 that they took. And that was followed by actinobacteria, acidobacteria, and Canidasis or Brocobacteria. They were relatively abundant, with being about 72 to 96% of those samples. The main ecological functions in microbial communities were chemoheterotrophy, methanotrophy, sulfur and nitrogen metabolisms, and CO2 fixations. Alright, so materials and methods. So I just went over that it's the La Palma Island, which is located in the northwest of the Canary Archipelago. This island shows a particular topography. There's 2 volcanoes on the island. One is active and one is not. This is from the active volcano. This is the cave system that they went into. You can't really see it, but these little orange blips up here and down there are the sampling areas that they took in 2015 and 2016. They revisited the top one in 2016, and the second one was what they added on in 2016. So, yeah. Okay, so for sampling, they had these different microbial mats listed in here. They refer to the coloring on these as if it's ochre, but it just means clay-like in color. So it ranges anywhere from these dark brown, orangey colors to this very viscous, mucus-like orange to this whitish color. Yeah. So those are the 4 areas that were sampled along the cave length. And so they also had this for scale on how long they were taking, and then that was for color matrix reasons. Okay, so DNA extraction and sequencing, phylogenetic analysis. So what I took from all of this was that they collected genomic DNA. It was extracted and quantified using a qubit 2.0 fluorometer. They focused on the V3 and V4 regions of the 16S rRNA gene using a 31F and an 805R primers in order to isolate the specific lengths of DNA that they wanted to use. With this data, they formed a bubble plot illustrating the data using our package, if anyone's familiar with that. And they also figured out the ecological role of these microbial communities. They predicted it with a bioinformatics software package that they ended up using. Great. So results and discussion. So for microscopy, they found a lot of actinobacterial-like morphologies similar to other volcanic caves. The microbial features compromise like coconut-shaped and filamentous shells with cells with spiny surface orientations. And in the clay-like stalactite, they found a lot that were probably actinobacteria based solely on the looks of them, which I think is in the next section here. Yeah. Let me see that a little bit so you can see it. So these are the different field emission scanning they were doing. So, but yeah, this is in. But yeah, so A and B are the clusters of actinobacteria. C and D ended up being no, C was some more actinobacteria from the soft stalactite that they ended up using. So these are just the different parts of it that they ended up doing. So I'm going to explain these tables real quick. So these tables, they were for the microbial community composition. So the Simpson-Lundy index, which is here, shows when there's a relative abundance of species where values range between 0 and 1. With samples close to 1 is when samples are close to 0, when no diversity is observed. Whereas whenever it's close to 1, there's a high diversity observed. So in this third sample, they ended up with their highest diversity at 0.993. Most of them were pretty close, other than this last one, which had only a diversity of 0.706, which still is pretty high, but it's not as high as the others. And so the Shannon-Weaver index focuses on species richness, and the value increases when the number of species also increases. And it also, the individuals among species becomes even. So the highest one is this third sample. The highest is this third sample with this 10, followed by the second sample, followed by the first sample, and then last but not least, the fourth sample. But yeah, this is based off of the outright diversity for the 16S RNA, as it says in that. And so a lot of this ends up just going into very fun, nitty-gritty of how each of these things interacts with each other, and a breakdown of each of these things. And you see there's a ton of protobacteria here in all of these samples, having the highest amount in each of these. Okay, that's this page. So the heat map also revealed, which I will talk about next, which is this. What little time. So the heat map revealed that there's a dominance of gamma protobacteria over all other classes, followed by actinobacteria, and there's also a relative abundance of protobacteria. You can see, so the abundance is shown with a very dark blue color. You can see with the gamma protobacteria just had the highest amount for this at class level. And then we have another heat map, which is at the genus level. So we have this one right here, which is just a uncultured bacterium, ends up being the highest for genus level. So this also highlighted during this part that the there we go. Sorry, my bad. So it has that this class NC10 of Connectus Rocubacteria was the third group in abundance. That group has methane oxidation and anaerobic conditions, and so it's associated with nitrate reduction, and they had a pretty decent amount of abundance in each of these samples. So a lot of this ends up going into a lot of talking about each of the genus breakdowns and why they thought that it was like that and how it relates to other things. Like it has this one where it's talking about limestone caves, and this is not a limestone cave, it is a volcanic cave. So they were like, yeah, that makes sense why it's not there. Regarding the participation of bacteria in the geochemical cycle of elements, some insights are derived from taxonomical groups retrieved from the cave. There's a total of 54 microbial functioning groups corresponding to 19% of the OTUs that were identified. Within those identified, there was a high proportion assigned to aerobic chemoheterotrophy, nitrification, and methanotrophy across all of the samples. The major predicted ecological function was chemoheterotrophy, which were involved in the phyla of the protobacteria. Remember the graph that I showed you? That was one of the main organisms going on at that part. So this fun little graph right here, so it has each of the 4 samples labeled with the relative abundance in corresponding with the size of these circles and how each of the different of the 4 samples broke down. So mostly it went with chemoheterotrophy, then followed with aerobic chemoheterotropy, and then some nitrification, nitrate oxidation, and all that stuff. There was a couple of the time analysis down there, but mostly it stayed up here with their relative abundances being upwards of 40 or 30%. So this whole breakdown is on the different sections of DNA that they went over. The one that was in the most abundance was this 6412 acetyl-CoA carboxylase. That ended up being a pretty high spike in all of them, which makes sense for cave because it helps with nitrogen fixation. So first up is sulfur metabolism. So we have all these protobacteria in the samples that were shown. They contain sulfur oxidizing and reducing lineages. So in these genes, the cis-I and cis-J were known to be involved in the some pathway that synthesizes hydrogen and sulfide from the sulfides. They were produced as beta alpha and beta subunits of sulfide reductase activity. So then they had CO2 fixation, where the gene related to CO2 fixation were reported across all of these samples. So the most abundant CO2 fixation pathways were the pit crust, were the CPB cycle and the TCA cycle. And this goes over all of the different ones that go into this that they found in that breakdown. That was the last round that I showed you. So then they also had nitrogen metabolism, where that was represented by the nitro. I don't remember how to say that. I practice it.
DCA.
Yeah. Okay. But yeah, and it had that with the nitrospira, which has the nitrite oxidizing bacteria. But yeah. And so this all goes over each of the, like, this one says like likely the primary cause of like calcium carbonate precipitation, and they just keep going over like each of like the nitty gritty details of them. So not super duper important at this moment for a general overview, but other mineral elements in the volcanic rock from the cave are iron and manganese. They have not a huge amount of them, but it was enough to be a note. So overall, the conclusions are basically the same as the beginnings. And yeah. Any questions?
Okay, actually, I will call you on the whole grading thing.
Because I just had breaking down like every single thing, so I was just like,
But what was the overall message that this paper was trying to deliver to its readers?
like there's a lot that goes into each of the like the chemical cycling, like there's more than just one thing that does everything.
Yes, sort of, but yeah, it is the whole idea that you've got all of these different elemental biogeochemical cycles, and then they were actually looking at one, was the diversity present
Yeah.
to enable all of these different metabolisms to occur, then with the requisite microorganisms, the nitty-gritty present, and then with the specific genes, were they actually capable metabolically? So, so, okay. So, anything else for Gabe? A couple of other things I wanted to also mention. So you had mentioned the R package.
Yeah.
Actually, this is something that we don't offer here yet.